I call it a connection between our small inhabitants, our lifestyle, and our health, because as you will see, we are all connected with each other. And first of all, I want to tell you what are microbiomes, because the term is often heard in the last years in the media and in the public. And actually, the official definition of microbiome is a collection of all microbial genes or all microbial genetic material in a certain sample, which is found in a certain environment. So it's a very, very general definition. It's not only human, it can be in animals, it can be in plants, it can be in any other possible environment you can imagine. Very often it's used with another term, which is called microbiota, and this means collection of all microbes in an environment. Why we make this difference? Because we don't know actually 99% of all microbes on Earth, we still don't know. So basically, we so far we only characterized about around 1% or less than 1% of all microorganisms, and that's why the definition collection of genes or genetic material is more precise than the definition of the microorganisms, because we still don't know if a particular gene or protein or whatever belongs to a particular known species or an unknown species. Yeah. So everything happened because of the very fast and actually boom in development in technology in the last 15 to 20 years. We have seen the development of new sequencing technologies, especially DNA sequencing technologies, and all other technologies in molecular biology, in bioinformatics, in data science, and so on. I would compare the technological leap as big as discovery of microscope, because in the 17th century, when Anthony Leeuwenhoek first time saw his microbes under microscope, it was like, we knew that there's something there. It's not just the ether or air that is causing disease. We now know that actually there are many, many more species out there. And with these technologies, actually, we are able to see theoretically each microorganism in each sample we take. So basically, any sample you take, you can see all microbes which are there. As I say, we still didn't characterize them, but based on the bioinformatic technologies and other technologies, we can recover the possible genomes and possible organisms from a sample. So also important to say is that microbes are not only in the gut, because we often hear through many nutritional scientists or other media, it's mostly gut microbiome that is talked about. But actually, human microbiomes are all over our bodies, on our bodies, and in our bodies, right? So estimated numbers of species on our skin are more than 600, and different populations of microbes are different between different parts of our skin, for example. Then also we have microbes in our oral cavity, in our nasal cavity, and even in our respiratory pathways, which since a couple of years ago most people thought are sterile, but basically they are not. Even in a healthy state, they still contain a certain amount of microbes. As I mentioned, gut is the most researched, and most of the microbes in the gut are actually in the column, in the big gut, in the rectal part, and the column. And estimated a number of species varies between different researchers, somewhere between 500 and even 1,500 different species, which, as I said, they're not all characterized. Urogenital tract, when we say that, most of the species that has been analyzed are in the female reproductive tract, in the vaginal microbiome, but we also have microbes in our bladder or in other parts of the urogenital tract, which also have been thought as sterile since recently, and they're also important. Now we come to a question that everybody's asking: what means healthy microbiome? And actually, it's very difficult to define one microbiome as healthy, or pull a certain bacterial species as good or bad bacterial species. Also, the concept of good and bad bacteria has been changed in last years or decade, and now we know that some bacteria that previously have been thought exclusively as pathogenic are actually normal components of our microbial flora or microbiomes. Some things that actually we have to, when we think about microbiomes, we have to think about them as populations. Basically, we have to think in terms more of microbial ecology than the single species. And when you talk about microbial species in ecological terms, basically there is a term of resistance of a population. So we're talking on the level of population, basically how the microbial population or one's particular microbiome can resist environmental changes. And the other question is, how quickly is this population able to recover after, for example, it has been attacked by an antibiotic or something like that. And this is one thing. And the other thing which is also important to think about is actually that these species are in balance with the host. They have been co-developed with the host, and they live in symbiosis with us since we are here. Even they're even much older than we are. Eubiosis means a balanced state, where dysbiosis is usually defined as disbalanced interaction, which means that certain microbial species or certain groups of microbes are in a higher amount competing with others, and then can eventually lead to a disease. What we also learned in the last years through different studies of microbiota is actually that the diversity of microbes is very important in the microbiomes. And we also learned that, for example, in human populations, like indigenous human populations, for example, in Africa or in South America, which has not been in contact with the modern civilization, the microbiota is much more diverse than in our microbiota, particularly in the human population that is in the Western diet. So there is definitely connection between our lifestyle, our diet, and our diversity of our microbiota. But still, just defining the structure and saying we have particular species is not enough. So the other thing is, you can say, okay, maybe we don't look at the structure. We can look also at the function. So because, for example, there are different microbes which are having similar functions. For example, they can produce similar metabolites, but also that is not enough. And we have to look actually at the level of interaction with the host. And this brings the whole thing to a much more complex level, where we have to talk about the individual differences in genetical differences, metabolic differences, lifestyle differences, and then the differences in the individual microbiomes. So these are all things that are just at the beginning of research, and in the coming years we will probably learn more and more about it. And now I will just say a few words. This is another chapter in the book which is talking about an important point, which is a micro interaction between the microbiome or the connection between the microbiome and our immune system. Simple question is, why we don't get sick of microbes, because of our own microbes, because we know that our immune system usually recognizes the foreign body or a foreign microorganism and then attacks that organism and destroys it, or develops an adaptive immune response, which means it remembers the contact with the microbe, then we become immune to a certain microbe, so it cannot attack us. This microbe cannot attack us again. However, we live, as I mentioned, with the hundreds and hundreds of microbial species in and on our body, and why these microbes do not cause the disease. And simply it's because they develop together with us, and since we are born, we inherit a certain number of microbes from our mothers. Then in the first years of life, our microbiota develops. Basically, by the third year of life, babies usually have the microbiota which is similar to an adult. And in this period is actually crucial for the proper development of immune system. And experiments in animals, for example, there are germ-free animals like germ-free mice or rats. The experiments have shown that these animals they have severe abnormalities in the development of the immune system, even the brain development and so on. So one thing actually that is protecting us from inflammatory reaction, actually from the reaction of our immune system, is the gut barrier. So in our gut, there is the wall of our gut, and here's the illustration from the book on the right side. And our gut actually is covered with a thick layer of mucus of slime, and actually below that layer there is another layer of cells, and these cells are actually connected, just as the bricks in the wall, they're connected with the intercellular material, and basically this barrier prevents foreign molecules or unwanted molecules to enter our bloodstream. And other thing is actually this barrier is actually preventing the microbes to come into contact with the immune cells, and these immune cells are basically in the gut wall. And then if there is a damage, let's say in the mucus or in the gut wall, or this gut wall barrier becomes a bit leaky, then the foreign molecules can enter our bloodstream, and even microbial cells can pass between the gut cells and come into contact with the immune cells. The consequence of that is inflammation. So basically, then the immune cells immediately they react, they produce molecules signaling inflammation, and then this inflammation can cause a local inflammation. This can be a local inflammation. This can cause also systemic inflammation. If these molecules come in the bloodstream, then they go to the brain, and they go to the other organs, and this can cause the systemic inflammation in the body. And this inflammation doesn't have to be a severe one. Let's say that, like if you get a bacterial infection, it can be very, very low level inflammation. But the inflammation on this low level, if it lasts for a long time, can lead to disease. And so far, there's a whole list of diseases. The cardiovascular diseases, the cancer, rheumatoid arthritis. There's a whole list of diseases that has been actually connected to the damage of the gut wall barrier. We don't have time now to go into details. You can read more about it in the book if you're interested. It has been detailed there. And also another important part is that there is a connection between our microbes and the brain. Basically, microbes produce chemicals that can directly go and interact with our nerve cells, or can directly go to our blood stream. On the other side, there are special cells in the gut wall. Actually, these cells are specialized and to produce chemicals that then actually act as neurotransmitters. So these chemicals basically can then act on the endings of our nerves, and then the signals are transported to the brain. Or these cells also produce other chemicals that also are then secreted to the bloodstream and then can act on our brain. The second thing is also immune system, which is connected to our brain on several levels through different chemicals produced by the immune cells. So there are basically these three levels, like a nervous system, the blood system, and the immune system. These are three different layers or three different pathways, so to say, of communication between our microbiota and our brain. And so far, it has been shown that actually, besides different psychiatric conditions, there are also other neurodegenerative conditions, and there's a whole field of research now about how we can, modify the gut microbiota, for example, to modify also our mood, or even neurodegenerative conditions and diseases. And then also our complete well-being basically depends on the microbiomes. So one example, which I didn't mention, one of these chemicals, which is produced by these specialized cells in the gut wall, is serotonin. And serotonin is a very important neurotransmitter and hormone in our body, and it heavily affects our mood and behavior, and also proper functioning of most of the organs in our body. And more than 90% of the serotonin is actually produced in the gut. and only less than 10% elsewhere in the body. So basically, the interplay between the gut microbes and the serotonin producing cells in the gut seems to be very important. Another thing is also research on how the physical activity or exercise or sport influences the microbiota. One thing I didn't mention actually about the sleep and the mood pattern. Basically, the circadian rhythm also influences the microbiota. Basically, the microbiota are adapted. The structure of the microbiota changes with the circadian rhythm. So it's also our sleeping pattern also can influence the microbiota. Exercise, besides, it has been shown that actually the exercise makes the gut wall barrier stronger and has overall good effect on the body. Basically, there is now a larger field of research on the specific bacterial strains or differences in bacterial strains between people who have more like sedentary lifestyle, people who have like a moderately active lifestyle, and people who have, for example, very active lifestyle of professional sportists. And there have been certain strains that have been characterized in professional sportists or very active people, and there is a still ongoing discussion if these strains are actually due to the metabolic changes in the body. For example, if you run a marathon, your body will produce extreme amounts of lactic acid, and then these bacteria are there. We still don't know if these bacteria are there because of metabolism of lactic acid, or actually they help us to increase the metabolic activity. So experiments are ongoing now with these specific strains to show if eventually we can boost the endurance or performance, which will be very important for the sportists. Then, of course, the diet or the nutrition is a huge field of research, especially, and we focused on microbiota. And there is like a large amount of research showing actually that the specific bacterial groups have different effects on our energy metabolism. Basically, there are two big groups. I don't want to bother you with names, but there are two big groups of bacteria, which actually one of them is consuming energy very quickly and producing also a lot of energy, while the others actually prefer more like food which is not that high in energy content, and also producing less of energy. So the first group, actually, if it is dominant, if it's like, let's say, more than 40–50%, then actually it can lead to metabolic disorders, and especially, for example, it's found in obese people that actually they have different ratios between these groups of bacteria. Now I just shortly will talk about the applications of microbiota or microbiomes. One thing which I just mentioned is the nutrition, and the trend is now the current trend is actually so-called personalized nutrition, because, as I said, technology allows us now that we can in a very simple way, and even relatively cheap. Now you can actually characterize many microbes or the structure of the microbes in the gut. And there are many companies, well, not many, but dozens of companies, let's say, who provide now this gut microbiota testing, and then you get your results either via your smartphone or your computer, and then they can give you the nutritional recommendations. There are even more advanced tests where actually the blood test or even genetic tests are combined with the gut tests, and then they can give even better predictions of which type of food is for a particular person good for stimulating the beneficial microbes, let's say, in the gut. Also, you all know about probiotics, and I'll just make a short overview of the different things which are sold there on the market. Basically, there are probiotics, which are living strains of microbes. One important thing to say is that actually our repertoire of probiotics is quite limited, because there are only maybe around 200 or a bit more than 200 bacterial strains which have been officially licensed as probiotics, because we know that there are bacterial species, but on a level of one bacterial species, there can be many different strains. For example, just take a simple Escherichia coli, which is like a typical gut bacterium. There are many strains of E. coli, and some strains are good. I mean, good in terms of producing beneficial metabolites, while the other strains are actually even pathogenic. The same thing is like, for example, with bifidobacteria, which there are many strains, and most of them are beneficial, but some of them are not. And then we have also other category, which is called prebiotics. And prebiotics are basically the nutrients that are used by the beneficial bacteria, and then these beneficial bacteria will produce other metabolites, for example, like short-chain fatty acids or other metabolites, which are then beneficial for our body. And these are usually mixtures of prebiotic foods, let's say fibers or oligosaccharides, and the probiotic strain. So basically, it's like a formula of one particular strain or a mixture of few strains and the food for that strain. Of course, we can also stimulate it by normal nutrition, but this is kind of like a targeted approach with a certain probiotic strain. Then we also have another category which is called symbiotics, and symbiotics are basically a mixture of this is not what I mentioned now, the mixture of pre and probiotics. Exactly, prebiotics I already mentioned. And then we have postbiotics. Postbiotics are dead microbes, or like molecules produced by these dead microbes, which can have beneficial effects. These are just few categories, and current trends are also in this field towards development of next generation probiotics. And there's a large field of ongoing research, like worldwide, where now there's like a characterization of new strains with potential probiotic role. And there is also like a big field of there are a lot of problems within the regulatory field, because in some countries you can use them as probiotics, in some other countries you cannot. And also there will be also now probably a lot of changes in the future in the regulatory terms. Okay, the next big field of application is therapy. So the therapy can be divided in different sections. We have bacteriophages, and bacteriophages are quite interesting. They're actually viruses which attack bacteria. Basically, it's from the Greek word bacteria and phage, yeah, so phagus. So basically, they eat bacteria, bacterial eaters. And then actually, for each bacterial species, you have tens of different phages that can kill that bacteria. And we still don't know all of them, but so far there has been several companies in particular in the United States. In European Union is still not allowed to use live phages as a therapy, but I think that will change probably in the coming years. You can only use phage products like proteins and so on, but not the live phages. And then the bacteriophages actually are very efficient against bacteria, for example, which are resistant to the antibiotics. And as we know, our repertoire of antibiotics is getting very thin now, and there are many bacteria which are resistant to multiple antibiotics. And in this sense, bacteriophages are very, very efficient. And then we have also the field of food safety, which basically there are already patents which are used for food safety, where basically the food that is before packaging, for example, the food is sprayed with the phages and it cannot get spoiled with a dangerous bacteria. Then we have also another field of microbiome therapy, which is microbiome transplantations, and the most popular is fecal microbiome transplantation, or shortly called FMT. And this is basically very efficient in cases of people who have severe bacterial infection, for example, with Clostridia or Clostridium difficile. This is very dangerous, even in life-threatening bacterial infection. especially in people who got multiple rounds of antibiotic treatments. And then actually basically the whole microbial population in the gut is killed by the antibiotic, and only one species becomes dominant. And then by the FMT therapy, these people can be saved. And there are also several clinics or even companies now who try to make now new formulations. There's also discussion about the safety issues and so on, but actually it exists in several places in the world, the standardized FMT therapy. Then also the same principle is now explored for the vaginal infections, or dysbiosis, and also for the skin infections and so on. And then another very interesting field of therapy is actually so-called pharmacomicrobiomics. Basically, certain microbes are able to metabolically convert and modulate the effect of the drugs that are used in human therapy. And one particular example is cancer therapy, where actually it has been shown that one of the so to say modern cancer therapy is so-called immune cancer therapy or immune checkpoint inhibitor therapy. And in this therapy, actually, it has been observed that certain patients respond much better to therapy than the others. And then actually, it has been shown that there is a huge difference in the microbiota between these patients. And there are several companies also now trying to develop the preparations like a probiotic basis, or like a mixtures of microbes, or even for the transplantation therapy, that can be used as a co-therapy with another therapy, for example, cancer therapy. Then another field of application is diagnostics, and we have two different things. We have biomarkers. These are single species. Like, for example, if you have a tuberculosis, then there is like a Mycobacterium, which is like a typical species causing the disease. Or if you have a Helicobacter in your stomach, this also can be a bad prognostic marker for the gastric disease. But on the other side, we also now can see the mixture of molecules. We can see also like a specific mixture of genes or proteins or metabolites produced by the microbes, which not necessarily come from one microbe. They can come from several different microbes. And this mixture, it can be actually a predictive for a disease. And these are called microbial signatures. This is another thing which is also now, in development in the field of diagnostics. So just briefly about the future, the sequencing technologies are developing super fast, and we are seeing now small devices we can put on our palm of our hand, which can sequence huge amount of DNA in a short time, and new methods are appearing every day for the analysis of data. We have new machine learning methods that can even better predict if a particular species is there. Then there are the algorithms basically that assemble the genes from the sequencing. And then also, as I mentioned, there will be new regulatories that will be developed probably now for the new microbiome drugs or therapies or diagnostics or prevention in terms of probiotics and so forth. And also a very important thing, which actually my company is doing, and I'm trying actually to do as much as I can, is basically to raise the awareness about the importance of microbiomes. And this is why the company is called Microbiome Power, because actually we try to write the blogs and to write the articles, and also helping other people with writing text, and also for all kinds of audiences, from general audience to like a semi-specialist who need maybe more instructions or manuals or reviews, to like a full specialist in the field who need help on a specific topic. And at the end, I just want to thank you for the attention. And you're welcome to check and buy our book. It's available on all major channels, and also you can visit our website, microbiomepower.com, and you can contact me anytime, either via email. Here's my personal email, but you can also contact me through this info@email. And thank you for attention once again.